Welcome, everyone. It's a pleasure to introduce Luke McKay, this week's C-DEBI network speaker. Luke received his Bachelor of Science degree from the University of Alabama at Birmingham. His interest in the origins and limits of life led him to work with Andreas Teskey at the University of North Carolina, Chapel Hill, where he first pursued a master's degree in marine science by studying deep-sea microbial mats that colonize active hydrothermal vents in Guaymas Basin in the Gulf of California. After completing his master's, Luke started his PhD in the same lab to investigate patterns of microbial biogeography in high-temperature sediment. His most recent work focuses on the upper thermal limits of uncultured life in hydrothermally-altered mud. Luke became a C-DEBI graduate fellow in 2012 and successfully defended his PhD last week. So please join me in sending him your congratulations, and welcome aboard, Luke.
Thanks very much. Thanks, Steve. And thanks, everybody, for having me and joining in. I really appreciate this opportunity. Very cool to be a part of this. Yeah, so the title of my talk is High Temperature Microbial Life in Guaymas Basin Hydrothermal Sediments. And let's get started. So here's a map of the Pacific Ocean, and here you can see the East Pacific Rise plate boundary traversing across Pacific and jutting right up into Mexico, where it's separating Baja California from mainland Mexico, and thereby creating the Gulf of California here. So here's a close-up of the Gulf of California, and you can see Guaymas Basin is right here in the smack dab in the center. Here's another map of the Pacific Ocean, but this time I'm showing you surface water chlorophyll A concentrations. And as many of you, I'm sure, know, there's higher productivity in coastal areas, as you can see by these darker green colors. And higher productivity often comes hand in hand. That means higher sedimentation rates. And so here in the Gulf of California, there's very high sedimentation rate from surface water productivity, meaning that the spreading center is actually located beneath 300 to 400 meters of organic rich sediments, which makes Guaymas Basin a pretty cool, unique spreading center. And here is another picture of what this is now a cross-sectional view of the sediments in Guaymas Basin. And what you can see here is these diagonal lines would represent the sediments. And this here is an intrusion, an intruding magmatic sill. And as it pushes its way into the sediments, it's reducing the porosity of the sediments and driving the pore water fluids out of the sediments and towards channels or fissures where the fluids can make their way up towards the seafloor. And so as these fluids are coming up from the subsurface towards the sediment-water interface, here you can see this would be a zone where there's this active advection or diffusion of hydrothermal fluids coming up. And there's a microbial mat colonizing the surface here. And here it's just showing all of these carbon and energy sources coming from deeper below. And basically, that supports a thriving subsurface community that may be performing methanogenesis, anaerobic oxidation, methane, nitrification, sulfate reduction, sulfide oxidation, just to name a few of the many physiologies that could be going on down there. So this is actually this is a picture that was taken from the Alvin submersible of a orange and white Veggiatoa mat. And this is the temperature profile being measured in the mat. And the cool thing about these are they colonize the surface of active seeps. And so when you're scanning the ocean floor, you can use these colorful Veggiatoa mats to predict sites of thriving subsurface microbial communities. And so here, as you saw in the previous slide, that was a temperature profile being taken. And this is actually a collection of all 113 temperature profiles that were measured in 2008 and 2009 aboard two different cruises to Guaymas Basin. And on the y-axis here, you have depth, in centimeters. Note that's centimeters. This is very shallow. And on the x-axis, you have temperature going from 0 to 200 degrees Celsius. And we can see here is that most of the temperatures start from seawater values. The bottom water in Guaymas is about 3 degrees Celsius. And then if they're very inactive sediments, they may not increase at all and just maintain that 3 degrees Celsius bottom water temperature all the way down. Whereas in very active sediments, they may increase all the way to 200 degrees Celsius. So all that to say, the shallow subsurface thermal variability is extreme in Guaymas Basin. And this allows the investigation of microbial community that spans the gamut of psychrophilic to hyperthermophilic life. It's a really, really cool system. So now showing again the same cross-section schematic of what's going on in the shallow subsurface. And see all these carbon and energy sources, but if we overlay this sort of thermal gradient, you also see that it's kind of like a bug zapper. There's all these good, delicious energy sources down deep, but the closer you get to them, the hotter it gets. And so my big question was, how does this limit or structure microbial life in Guaymas sediments? And so first I want to look at what we know about temperature controls on microbial life. And this graph represents or shows us how Ken Takai and colleagues in 2008 have sort of set the upper thermal boundary condition for microbial life in a laboratory at 122 degrees Celsius, where this sort of burgundy line here represents a proliferating cell population at 122 degrees Celsius. And now this is a table of what we've found in nature, and this is obviously not a comprehensive, exhaustive table, but this is just a few things I've been able to collect. And actually, if anybody has any other studies of very high temperature life that has been found or implicated in nature, that would be really cool if you could email those to me. So yeah, right here is a list, and from what I've been able to find, the highest temperature at which life has been implicated is 91 degrees from a hot spring in Yellowstone, and this is a study by Dave Stahl and from 1985, and they did some 5S ribosomal RNA phylogeny. So this just kind of goes to show that not only is our investigations of high temperature life limited in the natural environment, even when we do it, we haven't done a lot of detailed 16S phylogeny at very high temperatures. So yeah, what's all this talk about 16S and RNA? And so RNA can be an indication of viable microbial life. And here, this cool spinning image is a diagram of the 30S small ribosomal subunit that prokaryotes have in their ribosome. And you can see this sort of peach color is the ribosomal RNA, and the purple is the ribosomal protein. And they're wound together, and the RNA component of that is the 16S ribosomal RNA. And so, first, RNA is generally less stable than DNA, and both are very unstable at high temperatures. So, analyzing ribosomal RNA instead of ribosomal RNA genes provides an additional safeguard to avoid false positive sequence-based detection of microbial life. In other words, it's better to look at RNA than DNA when you're looking at the limits of life. And then lastly, because we're looking at the 16S ribosomal RNA, it allows detailed phylogenetic investigation, so not only an indication of viable life, but also who's there. So this is sort of the research setup to answer this question, and this is a picture of 3 different mat systems at Guaymas Basin, and these represent cores. And so the plan here is to take push cores of sediments associated with the highest in situ temperatures and measure the geochemistry to look for evidence of carbon and sulfur cycling, and then perform RNA extractions, reverse transcription, and pyrosequencing of the 16S ribosomal RNA to detect and identify viable microorganisms. Hang on, my light just turned off, sorry. Okay, sorry about that. There are motion sensors. I'm not making enough motions, I guess. So, yeah, that's the plan. Here's the first mat. I've given these mats nice names to remember them by. This one's called Sunny Side Up because it looks like a fried egg with this orange Veggiatoa in the center and a white periphery. And you can see from the temperature profile that the temperature increases at depth down to just above 100 degrees Celsius, at depth being 45 centimeters below seafloor. And actually, this is first what I thought the temperature profile was, and this is from a one-time temperature measurement taken near the core, but it turns out I got really lucky, and we actually deployed a temperature logger for 8 days prior to taking this push core with the Alvin submersible. And the way these temperature loggers work is so they were deployed for 8 days in different mats around Guaymas, and they record temperatures at 5 depths every 5 minutes for 8 days. And here's the profile. So you can just kind of look at the first 2 levels, which were the 0 to 4 centimeter section and the 8 to 12 centimeter section, and see in this blue diamond shape here, this is the profile, the time series profile of that temperature at that level, and then 8 to 12 centimeters. It gets very hot, as you can see on this y-axis here, increasing just about up to 100 degrees Celsius. And actually, this section corresponds to one of the samples that I took from this core, which we'll talk about in a second. But first, I just want to say that the thermal fluctuations are very high in surficial sediments, as you can see from these profiles that are going all over the place. And the in situ thermal range may fluctuate by 25 degrees Celsius in less than a day, which you can see in this sort of peak here and then also here. So you can have these relatively rapid changes in temperature with these very steep thermal gradients. Here's another mat, mega mat, and this one increases. This one unfortunately did not have a temperature logger. I only have the temperature logger for the one core, so everything else I show in terms of temperature will be just one-time temperature profiles that were taken adjacent to the core just prior to the core being retrieved. So this one increases up to about 120 degrees Celsius at 45 centimeters depth. Then this one I'm calling yellow mat, and it was taken right here on the edge of this yellow Veggiatoa mat, and this wonky profile is actually a sampling error, but it takes a long time to explain. But if you have a question about it, I can explain it. Anyways, it increases down to or up to about 125 degrees Celsius at depth. And the last one I call the Eye of Sauron because it actually was taken within this mat, where actually it looks like the mat has maybe been blown to bits by how hot it is here, and it increases down to at depth about 185 degrees Celsius. So this one's very, very hot, but as you can see, it's an extremely steep gradient, and the surface of this is actually about seawater temperature of 3 degrees Celsius. So here are the 4 cores now, and so all of these profiles represent the first core from the Sunny Side Up mat, and in the first column you have all the temperature profiles, and the second column we have methane in millimolar in the black triangle on the top, and the stable carbon isotopes of methane in the white triangle at the bottom, and the same for DIC, the black square, and stable carbon isotopes of DIC on the bottom, sulfate concentrations on the top up here, and sulfide concentrations on the top or on the bottom down here. And so I sampled these cores just like this, and I didn't choose intentionally this weird sampling style. Actually was limited by how I was able to get successfully reverse transcribe and amplify RNA. So these colorful little lateral sections here are the successful samples, and you can make the assumption that for several depths, definitely the next deepest depth, and for several depths beyond that, I was unsuccessful and could not get RNA to amplify. So already we're kind of we're approaching some sort of a limit. But it's not always temperature dependent. As you can see, I was only successful in this core at the coldest depth. But yeah, they're all very hot cores, so there's some temperature limitation going on here. And so here's just a summary of the temperature, in situ temperature for each of those samples. And I'm reporting this one, if you'll notice, this one is the one I showed you before from the temperature logger. So this is reported as a range from 77 to 100 degrees Celsius. And that's really important, I think, because this was an 8-day range that those sediments experienced prior to when the push core was taken. And so this is really the reality of what's going on in the thermal regime for this particular sample. So this is my favorite sample because I have this awesome temperature data. So yeah, I now want to look at the geochemistry, and I want to preface this with the disclaimer that the interpreting pore water geochemistry in Guaymas Basin is severely confounded by the dynamics of the flow regime. So there may be a vective flow going on, diffusive flow, and there's a lot of things are acting together here, making it very difficult to interpret. So every time I interpret one of these profiles, there's going to be a question mark after it. And what you can see here in the methane isotope, the stable isotopic composition of methane in this first core is really the only one where there's this large deviation towards heavier values, and that's indicative of the anaerobic oxidation of methane. And it could be happening in this surface layer, in this middle layer, or down here, depending on how you interpret it. And really, that's the only place where that seems to be happening. And now, if we look at the DIC profiles, where they're getting uglier and uglier, but again, this top one's kind of nice, and we can see that there is some evidence of organic matter remineralization, which is sort of indicated when there are lighter stable carbon isotopic values for DIC due to the biological release of carbon dioxide during organic matter remineralization. And so what you can see here is there's pretty strong evidence in the first core, and then maybe some evidence in some of the other cores, but we really can't say much at all about that. And then now if we look at the sulfate and sulfide, we see that there's some strong sulfate reduction in every core, and maybe some sulfide oxidation, depending again on if you're looking at it going up or going down in some of these cores. So now I've got these the temperature and the geochemistry summarized over here, and we're going to look and see who's there. So in the first core, the top and bottom layers were dominated by bacteria within the proteobacteria and the firmicutes, actually. And the middle layer was dominated by archaea. And you can see these white dashed lines represent the divide between bacteria and archaea. And the reason I'm presenting it this way, I haven't told you yet, is because I used universal primers that simultaneously amplified the archaea and bacterial sequences. So these relative abundances are best viewed at the domain level with archaea and bacteria together. Then the following cores are all dominated mostly by the archaea. And so what you can see here, the first observation I made is that only 2 out of the 8 sediment layers were dominated by bacteria, the rest were dominated by archaea. But since you can't tell a lot from domain level phylogeny, let's take a closer look. And now we're just going to look at archaea first. And we can see that there are a lot of different types of anaerobic methanotrophs, or putative anaerobic methanotrophs, falling within, in this green color, the ANME-2D or GoM Arc-1 or Methanoperedenaceae, depending on what you prefer to call them. And here, this is a very interesting core, I think, because the geochemistry was distinct, and there was this very strong curve or gradient that was captured in the sampling regime, and you can see a change in the ANME group. So here we have ANME-1 Guaymas archaea that's sort of enriched, or possibly enriched in the middle layer. And then down here in the hottest core, actually, this was very surprising to me, is ANME-2C, which is typically associated with colder sediments. But again, this is the surface layer of the hottest core, where presumably the fluctuations, thermal fluctuations, are very strong. So I think maybe there's some ANME-2C that there might be an increasing temperature regime that they're not happy about, or maybe there are also some high temperature liking ANME-2C. So now if we look at the bacteria, we can see that in the hottest sample, it's really dominated by the Thermodesulfobacteriaceae, which is a hyperthermophilic sulfate reducer. And the surface, there are some firmicutes, which are actually some Clostridia, thermophilic Clostridia, or closely related to those, and a lot of Epsilonproteobacteria in this blue here. And so yeah, that's basically what the bacteria look like. And so some observations here: archaeal sequences dominated by anaerobic methanotrophs and relatives. Distinct ANME subgroups are associated with different microenvironments. Bacterial sequences dominated by sulfur cycling bacteria, and the highest temperature samples have distinct sequence assemblages. So here is an OTU network that I made out of these samples, and the OTUs were defined at 97% or greater sequence similarity. And what you can see here is the big circle with the sample names. Those are my samples, and the little white dots are OTUs. And so where one sample connects to an OTU that connects to another sample, that means that that OTU was recovered from both sample S1 and sample E1, like so. And I wanted to take a look at what are the most probable hyperthermophiles, because there's a lot of diversity in this data. And so I took the 4 hottest sediment layers here, and which are represented by these bar graphs here. And I basically made this OTU network, and what you can see, this is the same thing I showed you before, but now the colder samples are whited out, and only the hottest samples are in color. And so I was using this to identify co-occurrence of OTUs across only the hot samples, meaning this would be the most probable prediction of what a hyperthermophile is. And you can see there's one right here, there's another one here, and another one here. Can't really see those very well, but they're nearby those big stars. So when I make a phylogenetic tree of who those actually are, it appears that the most probable hyperthermophiles are OTUs related to ANME-1 Guaymas archaea and a deeply branching miscellaneous cranarchaeoidal group archaea, and for the bacteria, it's the Thermodesulfobacteriaceae. So that appears to be the most probable hyperthermophiles. And here, now the next question I had was, what actually controls the microbial richness and diversity in these high temperature sediments? Because the diversity was so different across different samples, it doesn't appear to be that controlled by temperature. But if we look at the Cal1 estimated archaeal richness, which here is just this one-dimensional spectrum where I've normalized the most richness as estimated by Cal1 sample as the furthest end on this end of the spectrum, Y1, and then the least rich sample is the furthest in on this side, and then everything else is distributed across this spectrum. And this is pretty cool relationship where it looks like temperature has very strong control on archaeal richness with the 4 hottest samples. So this is the same type of spectrum where now it's reversed with the highest temperature sample over here and the lowest temperature sample over here. So the 4 hottest samples have the lowest archaeal richness, and the 4 coldest samples have the highest archaeal richness. That's pretty cool observation. And it just shows that there may be this inverse relationship between archaeal richness and temperature. And so I took a look at the same exact thing, but now this is bacterial richness, and it appears that it is not structured by temperature like the archaea was. For instance, sample E1 is pretty cool, but it has fairly low, relatively low richness, and same for sample Y1 here. So I thought maybe this could be limited by electron acceptor availability, and also it doesn't appear to have a very strong relationship there either. For instance, sample E1 has a lot of electron acceptor available, but very low richness. And so yeah, it doesn't appear that temperature or electron acceptor are as strong of controls on bacterial richness as temperature is on archaeal richness. So now just looking at the beta diversity. So I know this is kind of a busy plot. This is a principal components analysis, unifrac generated principal components analysis of beta diversity within the archaea. And so what this means is 2 samples that cluster close together are very similar in terms of diversity, like sample S1 and S3 here. And the gray spheres are actually the 5 most abundant recovered archaeal OTUs. So you can see that the reason that sample S1 and S3 are very similar in diversity is actually because they both have a lot of Methanoperedenaceae in them. And this is pretty cool because it shows that the beta diversity is structured very nicely by the groups that were found within those samples. And if we look to see if these physicochemical variables impart controls on beta diversity, it doesn't appear that they do. You might expect sample E2 and S2 to be near each other in terms of beta diversity, but they're actually very different. And sulfate concentration, same thing. Sample S2 and S3 are close together here, but they're very far away from each other in this. And so actually, I also looked at this for bacteria, and you can see similarly these Epsilonproteobacteria are influencing these 3 samples to be close together. And again, the temperature, sulfate concentration don't impart strong controls on bacterial beta diversity either. And so I thought maybe, well, domain level diversity is maybe a little bit too broad. So I'm going to look more specifically at methane cycling archaea and deltaproteobacteria. And so this is, again, another PCA plot of beta diversity across methane cycling archaea, deltaproteobacteria. And don't see, so I'm only showing you temperature and sulfate concentration. I actually did this for all of these one-dimensional combinations. And nothing seems to explain the beta diversity seen either at the domain level or at the physiologically targeted level. And in fact, no 2-dimensional combination of any of the physicochemical variables. There were, this behind you, obviously you can't see this, but this is just 45 different graphs that represent all the possible 2-dimensional combinations. And so far I haven't found any multi-dimensional combinations that explain this beta diversity. And so I kind of realized I was sort of looking for the magic variables that explain, microbial diversity, but I don't think that exists really. And so then I got to thinking, well, maybe I can't see how the physicochemical variables actually structure beta diversity, but might be able to see how beta diversity structures the chemical environment. And in fact, if I look at the methane cycling archaea and their influence on the methane, as indicated by the del C13, you can actually see this nice pattern distribution. And that can be largely attributed to the groups that are associated with those samples. So yeah, that was pretty cool. You can see localized isotopic evidence of anaerobic oxidation of methane, which can be assigned to distinct methane cycling archaea. But again, this is actually really hilarious. There's this website going around the internet right now that's very popular, and it's called Spurious Correlations. And so here I'm showing the number of films Nicolas Cage appeared in the last 15 years, correlated nicely with the number of people who drowned by falling in a swimming pool. So all that to say, there's major, you need to interpret correlations very cautiously. But I think that having this knowledge that methane cycling archaea might impart a control on the isotopic values of methane is a pretty strong indication that could be going on. So this led me to develop a new hypothesis that diversity is more dependent on the original inoculum than the local thermal and geochemical regimes. And then each distinct source community is then shaped by the localized physical and geochemical controls. So in conclusion, methane and sulfur cycling microbes dominate Guaymas Basin hydrothermal sediments. Shallow subsurface microbes experience high thermal fluctuations, and microbial life was not detected above the fluctuating range of 77 to 100 degrees Celsius. Probable thermo and hyperthermophiles are members of the ANME-1 Guaymas and MCG archaea and bacterial family Thermodesulfobacteriaceae. Temperature may constrain microbial richness more so than carbon and sulfur geochemistry, but none of these appear to dictate microbial community structure. And lastly, the inoculum of a particular sedimentary microenvironment may explain beta diversity better than the local physicochemical variables. So with that, I would like to acknowledge my advisor and all the people at UNC that helped with some of this data. And some other data that I didn't end up showing was generated by Doug Rowe at USC and Matthias Sabel at University of Bremen and Dirk de Beer at MPI Bremen. And then Jen Biddle and Karen Lloyd also contributed a lot to this study. So I would like to thank the shipboard and scientific crews and the funding sources, and especially C-DEBI for having me. And yeah, that's all. So I'll take any questions.
All right.
Does
Yeah.
anyone have any questions? There we go.
All right. Jen has a question. Oh, Jordan has a question. I don't want it to get lost. In Guaymas, does the original inoculum come from falling particles? Yeah, so that's a great question, actually. I think that you have to define original, right? I mean, I think that there is some falling particles are definitely going to be one source of an inoculum, but also I think a lot of it is just this dynamic flow through Guaymas sediments, because these are such high temperature sediments, and they may just get, from time to time, you saw how the fluctuations can be very high, may burn out all the microbes that are there, and then those sediments will get colonized, I believe, this is another hypothesis, faster than the sedimentation rate. And so I think that there is a lot of this dynamic flow going on that's carrying microbes from one zone of sediments to another. And let's see, Jen says, how do the deep branching MCG relate to the hot water Crin group? I didn't see where they were on the tree, just curious. Yeah, so I'm looking into the MCGs right now. In fact, this was actually another very surprising observation. It looked like they were sort of distantly related to the MCG 15, which was unexpected, because I think that was that's from colder sediments. if I'm right about that. So I'm not sure exactly how they fall with, or how near they are to the hot water friend group, but I'm looking into that. But right now I don't have, because I use pyrosequencing and I have shorter sequences, I don't have a lot of phylogenetic information to work with. So I'm doing my best to give it a place on the tree. So Karen says, how close is your sequence to the Methanoperedenaceae? Do you think there's a role for nitrate in the methane cycle of Guaymas? Absolutely. It's actually very close, I would say. I can't tell you the distance exactly, but I think that, yes, Methanoperedenaceae was recently shown to be a anaerobic methane oxidizer in a syntrophic partnership with nitrate reducers. And I think that that's actually like a glaring wound of Guaymas work, is if there's no nitrogen data really from Guaymas, or not very much. So I think that that's definitely a next step that should be taken in Guaymas. Okay, Matt says activate my webcam. Let me see how to do that. Matt, I think it's behind the actual video, and so I can't click it.
You click the icon with the three white people and the three green people and yeah, you got it.
Okay, cool. Yeah, so see any evidence for biogenic methane at Guaymas? Karen says, you see any evidence for biogenic methane at Guaymas? Yes, we do, but it appears to only, the isotopic evidence of biogenic methane is only visible at temperatures below 25 degrees Celsius. And that's not saying that that there's no methanogenesis above that temperature, but just that it only outcompetes the isotopic signal of AOM below 25 degrees Celsius. And but indeed we do see a lot of that. We see things down to minus 70 per mil of del C13 for methane, which is way lighter than the range of thermocatalytically derived methane.
Hey Luke, still can't see. Make sure to hit allow for your video in case you haven't done that yet.
Sorry. There you go. Sorry about that.
Yeah.
Okay. And Karen asks, did I see any methanogens? Yes. So not very many. I mean, you know, half of the groups that you see, you don't know what they are a lot of times. I did see a few things that I'm pretty sure are methanogens, but not nearly as many of those as what people putatively, you know, assume are ANMEs. But I think that I probably know what you're getting at here, which is, could the ANMEs also be methanogens? And I think that that's very possible. Okay, Jordan asks, does sulfate concentration predict archaeal diversity as well as temperature does? ANME reducing, hashtag ANME reducing your unsulfate. No, it doesn't. So, and I would like to just state again that nothing predicted archaeal diversity. It was just archaeal richness. And so just the number of different OTUs in a sample was limited by increasing temperatures. But in terms of predicting how the diversity would, how beta diversity is different across samples, that was not predictable by the temperature regime. Jason says, can you tell us how you used your OT network to deduce putative thermophiles? Yeah, sure. So this was just, I've never actually seen this before. I just had the idea that that if I found OTUs only in my hottest samples and they weren't also in the cold samples, they would be the most probable thermophiles. So yeah, that's true. It could be Rosalind. Sorry about that. So yeah, I just looked at co-occurrence of OTUs that was exclusive to hot samples. And so one of my committee members actually suggested that I could use co-occurrence of OTUs across hot and cold samples to deduce where thermotolerant microbes might be, or which microbes might be thermotolerant as opposed to thermophilic. I think that might be a good idea, but again, this is all just in the absence of culturing and all of that you all know about. This is the best that I can get at for guessing at which OTUs might belong to hyperthermophiles. Steve says, how would the thermophiles get into the hot samples if they weren't in the cold samples? Well, I think that, first of all, they might be in the cold samples, but I just don't detect them. And I'm only detecting the most active groups, potentially, because I'm reverse transcribing and amplifying RNA, which can be, in some cases, correlated with microbial activity. Yeah, so they might be there, or like I said, when a new hydrothermal seep comes to be, and there are these dynamic flow regimes going on, I think that introduces a lot of new inocula to the system. And so it could be that they just swing in and there's enough mixing in the whole system to sort of bring certain inocula from nearby sediments to the local sediments. So I don't know. I think the idea of inoculum is really interesting in Guaymas, because you have sort of these controls that are pushing on it from all different angles. So I would like to look into that more. Okay. Almond Lab says, can you hypothesize the cause of richness correlating to temperature? I guess just that there are a few specialized archaea that can deal with fewer, I should say, specialists versus generalists, basically. So you have specialists that can handle the higher temperatures, and generalists that generalists and whoever else can handle the colder temperatures. I think that there's just a lot more stress at higher temperatures. So maybe that's why richness is limited at high temperatures. Thanks, Karen. Yeah, Tina, I did see some heterotrophs, but I think actually a lot of them are probably heterotrophs in the sediments, like the I think the sulfate reducers that are there, like the Thermodesulfobacteriaceae. I'm not sure if that one in particular is a heterotroph, but there's so much organic matter there. Yeah, exactly. There's so much organic matter that that's what's fueling a lot of these. So yeah. And Jordan says, yeah, MCG could be a potential heterotroph. That's another thing that's just across the board with this, is that we do as best we can to give physiologies, or give like metabolic tendencies to these recovered groups, but there can be a lot of variability even within very closely related, phylogenetically related sequences. So who knows? Yeah, that's certainly possible. Reactivity of organic matter controlling the community structure as well. I think that's definitely possible. And we did measure organic acids, a handful of organic acids, but I don't have that data. But I should probably look into that. Maybe that could answer some of those questions. There you go, Jordan got right to it. There, in certain sediments, I do know that there's very, very high acetate in certain sediments. I can't directly off the top of my head tell you which sediments in comparison to which sediments I've just shown you today. But I know that some sediments in Guaymas have extremely high acetate concentrations. Multiple attendees are typing. Uh-oh. Why do you think the cold communities looked so different from each other as opposed to just being deep water like? Yeah, that's a great question. I'm really not sure. I think, again, maybe this could be the evidence of sort of the particulate inoculum that's coming down, and it's just, again, it depends on that local inoculum of that particular sediment. I mean, there is some consistency in Guaymas, but in terms of which groups are found in which types of sediments, but it just wasn't seen in this particular study. And yeah, so I'm really not sure about that. But I will say that calling these cold communities is sort of dangerous because they're the surfaces of very hot cores. And so there's a lot, and like I showed you, there's a lot of thermal variability. And so that could have something to do with it because, again, there's a lot of stuff traveling through those sediments. Thank you. Steve says, how does the core top temperature compare to the water column temperatures? Yeah, so in certain cases, can you still see my talk? You can't? Okay. Well, in certain cases, it's very cold. It's very, very close to that. In other cases, like in the Eye of Mordor core, the gradient is so steep that it's almost immediately, it goes from 3 degrees Celsius to 100 degrees Celsius in a matter of 10 centimeters. So I can't really perfectly identify the thermal range of any layer with a lot of confidence, because if I'm off by a few millimeters, it's going to change the thermal range drastically. So that's why I think it's best to report these as thermal ranges, which I was lucky enough to have for one of my cores. So, yeah.
Hey Luke, your slides are visible now. I just moved the video out of the way, so if you wanted to point to it, you can do that.
Yeah, so you can see in most cases the seawater temperature of about 3 degrees Celsius is characteristic of the surface sediments, except for in this case where we don't even have the surface temperature for this. Thanks, Jordan.
All right, well, that looks like yeah. Oh, hold on. Yep. A bit more, maybe.
Thanks, Dan.
All right. Thank you, everybody. Yeah, so this presentation, Adobe Connect Ruling, will be put up online in a couple days, and stay tuned for our next network speaker, Jason Sylvan, coming up next month.
Thanks a lot, everybody. Thanks for coming. This was fun.